Hey everyone, this is Dr. Cummings from Point Loma Nazarene University, and we're going to continue with our chemistry review here in episode 2 of Microbiology of Infectious Diseases. In this video, we want to talk about carbohydrates, so we're getting ready to talk about different macromolecules, the 4 major macromolecules that are found within the cell. And we're going to start with the carbohydrates, which refers to both the monomers and the polymers of sugars and polysaccharides, and then we're going to look at some of their functions. So the macromolecules are the 4 major building blocks that we see that all cellular life forms are built on, and 3 of the 4 are true polymers, 1 is not. And if they're polymers, that means they have a monomer as well, and it's important for you to understand the chemistry and the structure of both the polymers and the monomers, as well as some of the basic functions. So the 3 that are true polymers are the polysaccharides, the nucleic acids, and the proteins. You can see what their monomers and their functions are there. The 4th that is really a separate set are the lipids, and these are diverse hydrophobic molecules with diverse functions. Basically, anything in the cell that is hydrophobic in nature, we're going to label a lipid, and there's no real polymer to lipids, no real monomer to lipids, and so they're not true polymers. We're going to start with this top one, the polysaccharides. Poly meaning many, and therefore saccharides being the monomer. A monosaccharide is a single, simple sugar. The main functions of both the sugars and the polysaccharides are going to be energy storage and various structural roles that we see within the cell and within larger living organisms. So if we start with the monosaccharides, these are sometimes called simple sugars. This is where we see the name carbohydrate come out, right? You see the formula CH2O, carbohydrate. When you break it down into its simplest form like that, it looks like it's hydrated carbon. Now, realistically, we don't see any H2O molecules in there, but that's just the ratio that it turns out to be. So a carbohydrate then is going to be either a monomer or a polymer of these simple sugars, and they're going to have this general formula of 1 carbon for every 2 hydrogens for every 1 oxygen, and the little subscript N tells us that there can be multiples of these. So for example, you see here glucose, C6H12O6. In this case, N equals 6, and you can see that ratio of 1 to 2 to 1, carbon to hydrogen to oxygen, is upheld, and therefore we have a carbohydrate. In the image on the bottom, on the left side, you see the linear form, and the linear form is relatively common in solution, and it's in equilibrium with a ring structure. The main functional groups that we see that define a molecule as a monosaccharide, as a sugar, are multiple hydroxyls. So if you look at carbons 2, 3, 4, 5, and 6, all of them have an OH hydroxyl group associated with them. And then a carbonyl carbon, that's carbon double bonded to oxygen. In some cases, such as glucose, it's a terminal carbonyl, and therefore it's an aldehyde, and we would call that an aldose. If it's an internal carbonyl, it would be a ketone, and we would refer to that as a ketose sugar. But you can see that when the ring structure is formed, we lose our carbonyl and we gain another hydroxyl in the process. But of course, we lose that blue hydroxyl from carbon number 5. The net effect is we still have a lot of hydroxyl groups. We lose the carbonyl. And so lots of carbon, lots of hydroxyls in the ring structure. In solution, we see the equilibrium between the linear structure and the ring structure, but when we crystallize sugars, we have entirely ring structure sugars. So monosaccharides, these tend to taste really sweet, high energy source. Glucose is arguably the most important source of carbon and energy in all cellular life. There's only a handful of things we know of that can't make use of glucose. Now, a disaccharide, as the name implies, is just when we link 2 of them together. We're going to link them together by a reaction called dehydration synthesis. And this is a really common general form of chemical reaction for linking monomers together. It's called dehydration because even though there wasn't water, so to speak, in the original chemicals, we're going to remove an OH from one and an H from another, combine them to form H2O, and water will actually leave. And it will leave behind, let me see if I can get a pointer here. Here's our OH and our H. It takes water away, that's why we call it dehydration. And then we're going to leave behind a covalent bond from this carbon here all the way across to this oxygen. And that's what we see here, from carbon number 1 to oxygen from the hydroxyl on our second glucose to carbon number 4 of that second glucose. And we call this a glycosidic linkage. This carbon to oxygen to carbon combination of covalent bonds between two sugars is a glycosidic linkage. Now your book's going to get into things like 1,4 glycosidic linkages. All that says is carbon number 1 is linking to carbon number 4 on the next sugar. Don't sweat that. You'll learn more about that in organic chemistry. And it can also get into alpha and beta terminology. Don't worry about that until you get to organic chemistry as well. But you can see in this case, we took 2 glucoses, 6-carbon glucose molecules, dehydrated them together to form a 1,4 glycosidic linkage, and now we have the sugar maltose. You're familiar with maltose, like malt balls or a malted shake. It's a kind of a sweet, chocolatey sort of flavor. The disaccharides tend to be the sweetest and the most flavorful of all of the sugars. And that's what we see in this bottom figure here. You've got a 6-carbon glucose. We dehydrate it with another 6-carbon fructose. In this case, we're forming a 1,2 glycosidic linkage between these two 6-carbon sugars. And we end up with sucrose, which is table sugar. This is the white crystalline sugar that you bake with, or maybe you put it into your coffee in the morning or something along those lines. That's sucrose for you. So a disaccharide is when we dehydrate 2 monosaccharides to synthesize them, to bring them together and create a glycosidic linkage. Now, we could talk about trisaccharides, but typically once we get past di, we start calling the rest of them polysaccharides for many sugars. This is just a polymer of more than 2 sugar monomers. Some of these polysaccharides have hundreds or even thousands of sugars strung together. And the patterns of which sugars come where, and whether it's perfectly linear or if there are chains of sugars branching off of the main chain, really determines the overall structure. So what do we use these polymers for? There are structural uses. So, for example, what you're looking at here are yeast cells. This is Candida albicans. This causes yeast infections in humans. And these yeast cells have a cell wall wrapped around them like a shell made out of a polysaccharide called chitin. Happens to be the exact same material that you see on the protective shell of a crab or a lobster. So imagine little single-celled microscopic organisms with these little chitin shells around them, and you've got a yeast cell. So polysaccharides, really important as structural components of cells, but they're also important as storage compounds for those sugars, right? Those sugars are linked together and can be depolymerized to release the individual sugars, which can then be broken down further. If it's in a eukaryote, they'll get broken down in the mitochondria. If it's a prokaryote, they're going to get broken down in the cytoplasm with help from some enzymes associated with the cell membrane. All right, let's hit the highlights from this quick overview of the carbohydrates. So the term carbohydrate refers to both the monomers and the polymers based on that basic definition of 1 carbon to 2 hydrogens to 1 oxygen. And the defining features that we see that make a molecule a sugar are multiple hydroxyl groups and a carbonyl, either an aldehyde carbonyl or a ketone carbonyl. The mono and disaccharides are really important carbon and energy sources. And then the polysaccharides are important ways to store energy as well as to build various structures. Okay, so that's all I want to do to review the basics of carbohydrates with you. And in the next video, we're going to look at the next of the macromolecules. So study hard, learn these well, remember them well, and we'll talk about them more as needed in class.